In this final section of our chapter on cell division and reproduction, our primary focus will be on meiosis. Meiosis is similar in nature to mitosis. However, there are some significant differences to illuminate. First, the parent cell is still going to be 2n, or diploid. In this case, we're seeing that 2n equals 4. However, in the human condition, we'll see that 2n equals 46. 2 sets of chromosomes, 23 different chromosomes equaling 46. The daughter cells for meiosis, because there are 2 divisions, we'll end up with 4 daughter cells. So you can see here that we have meiosis 1, in which we reduce the chromosome number. Here in meiosis 1 at the beginning, we have 2n equals 4. However, at the end, there are only 2 chromosomes in each of these cells, 1 of each homolog. So now the cells are 1n. In the second division, we see meiosis 2, we end up having another nuclear division in which the sister chromatids separate, and still we are 1n. So each cell now has 1 copy of each chromosome. And this is important because this is how we reproduce. You have to think about it here. We're producing sperm and eggs. And if a sperm that has 1 copy of each chromosome gets together with an egg that has 1 copy of each chromosome, now we have 2 copies of each chromosome in the zygote, which means that that zygote, or that embryo, is now going to develop with every single cell being back to this 2n condition, which again in humans is 46. So the number of chromosomes in human sperm and eggs is 23. That is 1 of each homolog. There are a couple of other special things that occur during meiosis, specifically prophase of meiosis 1. First of all, there's synapsis and crossing over. During synapsis and crossing over, the chromosomes will align in pairs. And as they align in pairs, they contact each other, and sometimes non-sister chromatids, that would be this inside one and this inside one, so they're non-sisters because they're not connected at the centromere. Sometimes these guys cross over. Here is the point of crossing over. Sometimes in this crossing over, pieces are exchanged, and that technically is synapsis. When the chromosomes pull apart during metaphase of meiosis 1, they've changed pieces. So now these non-sister chromatids are genetically distinct from chromosomes that were in the parent cell. Here's an example of what these chromosomes look like following meiosis 2, when they're each in their new nuclei. So there are 2 recombined chromosomes. Because of this crossing over event, we end up with greater genetic variation. So in essence then, it's the fact that we have sexual reproduction and meiosis that increases the genetic variation in a population. And this increase in genetic variation allows for much wider variety for natural selection to act upon. So again, as chromosomes line up during metaphase, on the metaphase plate, they could line up in a number of different ways. Here you can see the red ones are all on one side and the blue ones are all on the other side. However, they could have been in alternate positions, such as is illustrated here. That would result in, again, a different variety of daughter cells, as seen here on the left, and then a separate variety of cells that we see here on the right. So it's the events of both synapsis, and independent assortment that you see here that lends itself to this greater genetic variation in sexual reproduction. Now let's take a close look at an animation of how meiosis actually works. I know a picture is worth 1,000 words, but a moving picture is worth probably 1,000,000. So check this out.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis I, halves the number of chromosomes, and in the second division, meiosis II, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase I, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only 1 member of the homologous pair and will be haploid. In telophase I, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only 1 of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
So there you go, that's how it happens. Now let's take a look at some images of that and point out some of the key differences to summarize that video. In the top panel here, we have meiosis 1, and in the bottom panel, we have meiosis 2. The key points of difference that I would like to point out are that in meiosis 1, during metaphase, which is the best place for comparison, we see that homologous pairs are aligned on the metaphase plate. And then in anaphase, it's the homologous pairs that separate from each other. Then when we get to meiosis 2, you notice that it's individual chromosomes because homologous pairs have been separated, and each of the daughters from meiosis 1 now has one of each homolog. So it's individual chromosomes that align down the metaphase plate. And these individual chromosomes are composed of sister chromatids, so that during anaphase, we see sister chromatids separating from each other. So again, in anaphase 1, homologous chromosomes are separating from each other, and in anaphase 2, sister chromatids are separating from each other. This figure shows us some of the distinct differences between mitosis and meiosis. Again, meiosis is divided into two divisions. We have meiosis 1 and meiosis 2. In metaphase, homologous pairs align in metaphase 1. In metaphase 2, sister chromatids align, or single chromosomes align down the center. That's analogous to what happens in metaphase of mitosis. The end products of meiosis are 4 haploid daughter cells. So 4 haploid daughter cells. These could be sperm or they could be eggs. They each have 1 copy of each chromosome. Whereas at the end of mitosis, we have 2 identical daughter cells that are diploid, just as the parent was. So now let's take a look at the summary of a comparison between meiosis and mitosis, again in an animated form, because I think that really helps out.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
Hopefully that clears things up a little bit. Not every organism on Earth has the same sort of reproduction cycle. You can see here on the right side, we have a diploid life cycle, which is what we exist in. So we have individuals that in their adult life are 2N, or diploid. And then a diploid cell, a germline cell that's going to produce sperm or eggs, undergoes meiosis in order to produce gametes. The gametes, or the sperm and the eggs, are haploid, or 1N. In our case, they each have 23 chromosomes. Those 2 gametes, the sperm and the egg, come together during fertilization and sexual reproduction to form a zygote. And then the zygote divides by mitosis for the whole lifetime, producing an adult individual in a diploid form. Now, some other organisms have very different lifestyles. You can see that they'll have alternation of generations. In this case, the zygote is 2N, and the adult form is 1N. Now, this is a really interesting thought. How do you make sense of that? Now, the way I've always made sense of it is thinking of it like a campus. And on campus, on a regular campus, you have lots of adult individuals running around, well, maybe adult, running around. 2N human beings, and they meet up with each other and they sexually maybe reproduce, they get together. And then in the other situation where we have this haploid or alternation of generations, I think about these 2N zygotes as being maybe sperm and eggs running around campus. And the sperm and eggs get together and form a very small single-celled 1N individual that then produces gametes in order to grow into a 2N zygote again. I know it's a really difficult thing for us to understand only knowing our life cycle, but just be aware there are different ways that we can alternate between this 1N and 2N in sexual reproduction. Again, these are going to be found mostly in algae and in some plants, where the seed form, for example an apple, is a multicellular form. So now a quick look at what happens if meiosis doesn't go quite as planned. This is where we'll see some chromosomal abnormalities or anomalies. The first situation involves nondisjunction. In nondisjunction, we can see that chromosomes don't necessarily separate properly. This can either happen during meiosis 1 or it could happen during meiosis 2. Either way, you end up with daughter cells that have either too many or too few chromosomes. So in this case, we should have one red and one blue in each of these daughter cells. However, because of a non-disjunction event up here in meiosis 1, we end up having no red chromosomes in these two cells. Non-disjunction could happen in meiosis 2. In this case, you see again that we have some abnormalities in chromosome number within each of the daughter cells. Down syndrome is an example of this. You see trisomy, or 3 chromosomes, in the 21 position, so 3 of chromosome number 21. Tau syndrome, I think, is 3 of chromosome number 18. Either way, things go wrong. Most of these eggs or sperm result in zygotes that are spontaneously aborted or miscarried, especially with some of the more serious chromosomes, like chromosome number 6. If there's a trisomy of chromosome number 6, that fetus will not survive. And our bodies have a great sense to determine whether things are right and life is possible, or wrong and it's not possible. So spontaneous abortion or miscarriage usually happens for very good reason. It's because something isn't quite right. So, finally, let's take a look at what happens when synapsis, or the pulling apart of synapsis, doesn't happen in quite the way that it's expected to.
Of a chromosome or chromosomes. One way that this can occur is by a deletion or a duplication within one chromosome. A chromosomal mutation is a substantial change in the structure of a chromosome or chromosomes. One way that this can occur is by a deletion or a duplication within one chromosome. In the chromosome, shown here, the longer arm is labeled Q and the shorter arm is labeled P. Regions are designated with numbers. If a region is deleted, such as a large portion of the Q2 region shown here, then a deficiency occurs. There is now less genetic material in the mutated chromosome. A duplication occurs when an extra copy of a chromosomal region exists, such as the extra copy of regions Q2 and Q3 shown here. Both duplications and deletions can be created by abnormal crossing over during meiosis. An inversion occurs when a chromosome is broken in 2 places and reconnected so that a region is flipped from the normal order, such as the Q2 and Q3 regions shown here. Note that an inversion does not change the total amount of genetic material in the chromosome. Translocations involve 2 chromosomes. In a simple translocation, a single piece of one chromosome breaks off and is attached to another chromosome. In a reciprocal translocation, two chromosomes exchange pieces. Note that translocations do not change the total amount of genetic material. However, both inversions and translocations can lead to overall deficiencies and duplications of genetic material after meiosis.
So again, errors can happen during the crossing over in synapsis. If the crossing over happens at not an even place on the chromosome, so one arm's longer than the other arm, then you may have some genetic variation in that chromosome that's generally not good. But any of this genetic variation that we might see really is the foundation upon which natural selection works. Often the mutations are not so good, but sometimes they're favorable. So hopefully these lectures have been helpful for you in understanding cell division and reproduction. If you have any questions at all, feel free to communicate with me through the messages in the learning management system. I'm here to help you understand, so please don't be shy.